But the one thing that I'm going to be here to tell you a little bit about is what if you don't have to clear your brain in order to see in the brain? And that's what we've got here today, is UCI has invested in a preclinical and translational imaging center, and that's here to help researchers, particularly my own interest as well, brain researchers to investigate using MRI non-invasively, both in vivo and ex vivo, to sort of parse brain function. And first of all, it would be remiss if I did not thank the partners who have, over the last several years, contributed extensively to this effort. As you may know, MRIs are not cheap, and there's a lot of infrastructure that goes with that. And so I have to thank everybody across campus. And this is truly a cross-campus facility that can be used by everybody on campus. So again, many thanks to those partners and colleagues who have helped make this work. So what does this look like? So the instrument in the Preclinical and Translational Imaging Center, the first one, I hope, of many, is a 9.4 Tesla MRI. This is about 3 times stronger than the nice fancy fiber. And of course, with that, you get increased resolution and some other advantages. So it's a fairly large bore instrument that will be able to allow imaging for a variety of rodents, small dogs, cats, the cat that was going to eat the cat, or the mouse there, we could image that, for example. And of course, we can do lots of in vivo imaging, which is be its power, but we can also do things like MR and MR microscopy. So if we image for very long time periods, we can get down into the 75 to 50 micron range, depending on how much time and effort we want to spend on that. The other thing is that, of course, we have a variety of components that help facilitate this research. So we have what are called radio frequency coils. These can be designed or purchased based on your research needs. We have about half a dozen different kinds of these for various researchers. So we have ones for mouse, rat, and we even have larger ones for larger mammals. We also have microgradient inserts that allow us to get faster and better imaging sequences, and we can insert these as well as with these in combination and really try to improve resolution and quantitation. The next few slides, I just want to show you some of the capabilities of what this instrument can do. So for example, many of the things that we do are related to relaxation in the brain. So this is a rat brain here, of course, and it's T2 relaxation. You can do T1. One of the things that we'll be able to do, and I'm looking forward to this, is basically arterial spin labeling. We can look at how blood flow changes in the brain or throughout the brain. We can also do MR spectroscopy. So if there's a particular metabolite, for example, like glutamate or lactate or some other metabolic component that you're interested, using spectroscopy, we can actually go and parse this. And the nice thing about a high field, the higher field you get, the more clean these line widths get, and we can actually differentiate many of these metabolites. We can also do continuous ASL, just another type of arterial spin labeling. We can do lots of short TE kinds of imaging for anatomical structures. We can do angiography. With the right sequence, again, we can map out, and along with either contrast agents or without contrast agent, you can actually map out the cerebral vasculature in the brains of rodents. We'll be able to do what's called functional magnetic resonance imaging, which allows you to look at connectivity or regions of the brain that might be activated during a particular sequence. Let's say, pain or other kinds of things. We can use this to map this and then look at how different brain regions are connected together over time. And then we can also do fat and water imaging if you want to look at regions outside of the brain, for example, in the torso. And then one of my own favorites, of course, is diffusion tensor, which allows you to map structural connectivity between brain regions. And we've been using that with great success, and I'll show you some of this in a moment. And then finally, of course, there are cardiology applications. This is very daunting, of course, because the heart in a rodent is very, very, very fast. So this is not my favorite imaging modality, but if there's researchers on campus, we'll definitely work with you to try to do these kinds of imaging modalities. So I just want to give you, since everybody took the liberty of showing a little bit of science, I'm going to show you just a very tiny bit here, just some of our own work that we've done in collaboration with folks on campus. This is work with regards to febrile seizures with Dr. Tali Baram that we've done. And over the years, we've essentially done T2 relaxation imaging to identify a imaging abnormality that's located in the basolateral amygdala right here. If you see, this is a lot brighter here than it is over here. And the nice thing with T2 relaxation imaging, we've published a number of papers now where we're able to actually very cleverly identify brain regions that are highly susceptible for seizures in those animals that go on to have subsequent epilepsy compared to those that do not. So the nice thing, this kind of groundwork led to a funding of an R21 in children that we're now pursuing to actually pursue this signature in children. So that's a good example of translation. One of the other things that you can do is susceptibility-weighted imaging. We heard a little bit about iPS cells. If you were to incubate those with a contrast agent such as gadolinium or iron oxide or other kinds of things, you can inject these and then track them over time as they go and, let's say, invade an area of ischemia, as we've done. So we published a number of years ago on how to do this, and you can see actually, you can identify this loss of signal where these iron oxide cells have been invaded. You can also do the same thing in looking at beta-amyloid deposition in the brain. So amyloid oftentimes in many cases is associated with iron depositions, and here's the MRI. And this is a Prussian blue stain from the same animal, and you can see these Prussian blue stains. While we don't have the same resolution as microscopy, we can get certainly a very good handle on these components. And then also diffusion tensor. So this is a model of concussion. And you can see here, these are the different diffusion tensor metrics. We can get the T2, the fractional anisotropy, the apparent diffusion coefficient, the axial diffusivity, and the radial diffusivity. And if you look at first blush, many of these are not different. But with quantitation using atlas-based metrics, we can actually identify changes within the corpus callosum or the anterior commissure here and the corpus callosum using diffusion tensor in these sequential time coordinates. The other thing with diffusion tensor is, as you saw, we don't use the Allen Brain Atlas because the resolution is too high. So we use the different kinds of atlas that are basically set up for MRI. We can parse out cortical, limbic. We have an atlas that basically has about 80 different regions. And you can then kind of get these sort of heat maps after concussion type injuries, and you can look at basically how those changes over time occur between brain regions. We can also do what's called tract-based spatial statistics in these animals as well to identify white matter changes again. And then this was in MS model. And then here is essentially treatment modalities where we have a fluid percussion injury for TBI, and this is tractography. So after giving it a treatment with these alavanoids, what we found is that the white matter tracts were much more preserved in these animals than those that were not treated. So just in summary, again, we have MRI that allows you to do T1, T2 kinds of related relaxation, angiography, diffusion tensor, functional MRI, perfusion-weighted spectroscopy, and there's a whole host of many things. And one of the things that I think is a real strength of in vivo or even ex vivo MRI is that we have this sort of menu of modalities that you can go and then look for brain function and activity. And so I will close with just letting you know the instrument has been installed. We are ramping this instrument up over the next month or so, and we hope to be open for business starting January, February for routine imaging. So if you have any questions, we also have a table with a brochure out there. There's a slideshow going on for some questions. If you have any questions, let us know. If you're interested in MRI, you've never done it before, and want to know how to start, give us an email or call, and we'll get you on the road to doing in vivo imaging. Thank you.